Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullah. আমরা ডার্মাটোফাইটোসিসের উপরে একটা ছোট লেকচার নেব। ডার্মাটোফাইটোসিস Bismillahirrahmanirrahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Dermatophytosis এটি একটি superficial mycosis. Superficial mycosis also include dermatophytosis, pityriasis versicolor, keratomycosis, tinea nigra, black pietra, and white pietra. কিন্তু এই ক্লাসে আমরা শুধু dermatophytosis নিয়ে আলোচনা করব। ডার্মাটোফাইটোসিসের আরেকটা নাম হলো টিনিয়া অর রিংওয়ার্ম ইনফেকশন। এটার নাম টিনিয়া এবং রিংওয়ার্ম ইনফেকশন কেন হলো এটা আমরা পরে বিস্তারিত বলবো। এমনি বোঝা যায় রিংওয়ার্ম এর শেপের কারণে হতে পারে। This is infection of the skin, hair or nails caused by a group of keratinophilic fungi which are called dermatophytes. That is, they have affinity for dermatophytes, that is keratin layer of dermis. 3 groups of fungus are included in this group. Microsporum, they infect only hair and skin. Epidermophyton, skin and nail. And Tricophyton, so it infects hair, skin and nail, all 3 layers. Tricophyton has got 19 species and it infects hair, skin, nail, all 3 structures containing this keratin layer. Microsporum has got 13 species. It infects only skin, hair and hair. Epidermophyton, it infects only skin and nail. Dermatophytosis, that is a ringworm disease of the nail, hair, and or stratum corneum of the skin. And dermatomycosis means is more general name for any skin disease caused by fungus is known as dermatomycosis. Dermatophytes are keratinophilic, that is keratin-levic. Keratin is a major protein found in the horn, hooves, nails, hair, and skin. A ringworm disease, also called ringworm because herpes by the Greeks and nomos by tinea, which means a small insect or larva. Previously, it was thought that this ringworm disease is caused by a small insect or larva. So, why named as a ringworm lesion? Lesions on skin and sometimes nails have a characteristic circular pattern that was mistaken by ancient physicians as being a worm down in the tissue. These lesions are still today called ringworm infections, even though the etiology is known to be a fungus rather than a worm. And this is a classical ringworm lesion. Previously, it was thought it has been caused by a insect or worm. That's why it is known as tinea. Infections by dermatophytes, they can be of 3 origin: by geophilic species, keratin utilizing soil saprophytes, that is Microsporum gypsum. Zoophilic species, keratin utilizing on host living animal, that is Microsporum canis and Tricophyton verrucosum. Anthropophilic species, keratin utilizing on human, Microsporum odoni and Tricophyton tonsurum. So dermatophytes, they digest keratin by their keratinases. They are resistant to cycloheximide. So cycloheximide can be used in the dermatophyte selected media. They can be classified into 3 groups depending on their usual habitat. These three groups are anthrophilic, which on the host human being, geophilic on the livings on the soil, and zoophilic on other animal. So anthrophilic Tricophyton rubra, very common. Microsporum gypsum is geophilic, and Microsporum canis, cats and dogs. So dermatophytosis, pathogenesis and immunity. First there is a contact, and following contact there are some predisposing factors like trauma, moisture, crowded living conditions, and cellular immunodeficiency can lead to chronic infection. Reinfection is possible, but larger inoculum is needed and the course is shorter. Clinical classification of dermatophytosis according to the site involved: tinea capitis, ringworm infection of the head, scalp and eyebrow; tinea favosa, ringworm infection of scalp; tinea corporis, ringworm infection of the body, that is smooth skin; tinea cruris, ringworm infection of the groin; tinea unguium, ringworm infection of the nail; tinea barbae, ringworm infection of the beard; and tinea manum, ringworm infection of the hand; tinea pedis, ringworm infection of the foot or athlete's foot. This is a typical picture of tinea capitis. This is tinea corporis, tinea pedis or athlete's foot infection. This is dermatophytid reaction, this is allergic reaction. From this reaction, dermatophytes cannot be visualized by microscopy. This is tinea cruris or joke is tinea unguium or nail infection. Major sources of ringworm infection: school, military camp, prison, warm, damp areas, that is tropics, moisture accumulation in clothing and shoes, animals, dog, cat, cattle, poultry, etc. Let us discuss about the laboratory diagnosis of dermatophytes. It can be diagnosed very easily by direct microscopy. Treating this scraping from skin and nail can be taken, and snippets of the hair, and this is scraping and snippets, they are treated with 10 to 20% potassium hydroxide. Potassium hydroxide dissolves the keratin, not chitin, that is hyphae, is usually very effective in detecting dermatophyte hyphae in clinical specimens. The addition of chalcoflower white 1,4-polymer specific fluorochrome dye and dimethyl sulfoxide DMSO to the potassium hydroxide and viewing with a fluorescent microscope is it increases sensitivity. Besides this, lactophenol blue can also be used. This is the chalcoflower white stain. So its diagnosis is clinical. Then we can use Wood's lamp in the laboratory direct examination and can be cultured. This is when the hair is involved, there are ectothrix and endothrix. Ectothrix means outside the hair shaft, and endothrix means inside the hair shaft. Culture can be done in a specific mycological agar. Severous dextrose agar is non-specific. Microbiotic agar, dermatophyte test agar. And after their culture, they can be identified by colony characteristics and microscopic morphology. This is the structure of Microsporum species: thick wall, spindle shape, multicellular. These are the structures from their colony, microscopy of their colony, Microsporum species. Epidermophyton flocosum, you see bifurcated hyphae with multiple smooth glove-shaped macroconoidea. And Tricophyton species, large, smooth, thin wall, septate pencil-shaped hyphae. The left side one is the colony structure, and this is the microscopic structure taken from colony. There are other tests also, physiological test, special amino acid, vitamin requirement test, urea hydrolysis test, but we usually use the culture forming microscopy. Treatment, topical treatment includes miconazole, clotrimazole, iconazole, terbinafine. Oral treatment include glycophyllin, ketoconazole, itraconazole, terbinafine. Thank you very much. Thank you for listening.